Hello everyone, in this video we're going to discuss about proteins and its monomer, amino acid. So starting with proteins introduction and some interesting questions about protein. I want to tell you, okay, we all know that the monomer is amino acid and here is a question for you. I just wrote a H over here. So can you tell me protein is a homopolymer or is it a heteropolymer? Time's up. It's heteropolymer. Hetero means different, right? So the monomer of protein is amino acid, but if it's a heteropolymer, does that mean that there are more than one monomers of protein? No, it doesn't mean that. It just means that there are different types of amino acid. So if it's a heteropolymer, in a single chain of proteins, there are different types of amino acid. For example, it's alanine, it's a glycine, it's serine. Let's take it as isoleucine. So you see, there are different types of amino acid, which means that it's a heteropolymer. Next, is protein found in retentate or infiltrate? When we do the tissue analysis, if you have taken that video, so you must know what's tissue analysis. It's a experiment by which we can know that which biomolecule is a biomacromolecule and which is a biomicromolecule. So is protein a biomicromolecule or macromolecule? Of course, it's a macromolecule, which means that it's found in retentate. The most abundant protein in this biosphere is ribosco, which is an enzyme. The most abundant protein in animals' world is collagen. You must have heard of collagen fibers, right? So the most abundant protein in animals' world is collagen, and humans come under the word animal in terms of biology. So now is the time to talk about amino acid. So there are different types of amino acid, but only 20 amino acids can form protein, not in only our body, but in every living organism from humans to viruses. Only 20 types of amino acids can form proteins. Now moving to the structure of amino acid, it have 4 groups. And is this carbon a alpha carbon or not? Or in other words, is it a chiral carbon? I can't tell that right now because this group is not decided yet. I mean, like, okay, if I just write H over here, so is it a chiral carbon now? No, it isn't, because chiral carbon have 4 groups and they are different from each other. And in the structure of an amino acid, this part is constant in every amino acid. This is the amino group, which means NH2, COOH, which is carboxyl group, and a hydrogen. This is the radical group, and it varies in every amino acid. I wrote H over here, and this was the structure of glycine. Glycine is the only amino acid that is not a chiral carbon, which means it's optically inactive. Except glycine, every amino acid is a chiral carbon. They have a different radical group, not similar to these 3. So this is a basic information about amino acids. For example, the radical group decides the physical and chemical properties of amino acid. Alpha carbon or chiral carbon shows the optical activity, which means that they can bend light. All amino acids have chiral carbon, except glycine. Why? Because the structure, as you already saw before, but I'm just going to draw it once again for you. So this is the structure of glycine. So here two groups are similar to each other, which means it's not a chiral carbon, thus it will not show any sort of optical activity. Now these are some characteristics of amino acid. Amino acids are substituted carbons. Substituted methane. Methane is CH4, right? So if we substitute these three things, so we will get a amino acid, right? So that's why we call it as substituted methane. Then there is an amino group in amino acid, right? Now when this amino group is on the left side, we call it as L amino acid. We already talked about the L and D structure in carbohydrates, so you must know about isomers so that you can understand what is L and D. Now this is a capital D, not a small d and a small l. Okay, let me just explain it shortly over here. Small l and small d are for textile and levo, which are both types of optical isomerism, while capital D and capital L shows us the isomerism, which is stereoisomerism, which we already discussed in carbohydrates, and it is the type of optical isomerism, of course, but they're called enantiomers. Okay, I mean, enantiomers are mirror images, in other words. So in carbohydrates, we already learned about isomers. So yeah, this is that capital L. Don't mix the small l and small d with capital L and capital D. So amino acids are found in filtrate. Why? Because they are the monomers, right? While protein will be in the redundant. Only 20 amino acids will make protein. I already told you this. Then proteins are not directly made from amino acid. They are amino acids first combine together and form peptide, and when different peptides combine together, they form peptones, and when peptones combine together and modify and have a lot of other compounds, chemicals attached with them, they form proteins. Then the sequence of amino acid, the person who firstly observed the structure, the sequence of amino acid in insulin was F. Sanger, and thanks to him that we know a lot about proteins, because he discovered proteins and he discovered amino acid basically. So what he observed was that insulin is consisted of two chains which are combined together by disulfide bond, and in general it have 51 amino acid. One chain have 21 amino acids and the other chain have 30 amino acids, and in total they make 51 amino acids. So that's it for today. We did the introduction of proteins and a little bit about amino acids. Next we are going to discuss about the types of amino acid and how can you combine amino acids. I mean, how to form bonds between amino acid. Okay, here's another statement for you. The bond between amino acid is called peptide bond. Like carbohydrates have glycosidic bond, so proteins have peptide bond. Alright, that's it for today. Thank you so much for watching.